This is just regarding geeky services. This is demo class 1 regarding ASRB net, SMS, STO and ARS. So in this class, we are going to discuss like what are all the basic demo, like how the classes are going to be and what are all the things that we are going to concentrate. Like I will just give you a description like how we are going to start the classes and what are all the kind of things that we are going to do, that I will tell you in this demo. Regarding this NET, SMS, STO and ARS, the syllabus remains same for all the exams. That is a preliminary exam for SMS, STO, ARS for everything. So we divided the same syllabus only, but for our convenience and easy discussions, I've divided these chapters with the specific areas and the concepts, and reference books I have given. Somehow I will take classes on a daily basis, 5 days a week. And here we divided these chapters into like how what is this plant pathology, where exactly this plant pathology started, and again history. In history we have divided into mycology, bacteriology, virology, epidemiology, applied plant pathology, molecular plant pathology, biocontrol of plant pathogens, and various other kinds of, if there is any remaining leftover topics in the history, all those things that we will discuss. But the most important thing is every single day, for example, if the mycology was finished, on the next day exam on mycology, on the next day before conducting the exam, bacteriology will be discussed, and the same day exam on mycology, because the mycology was discussed in the previous day, and the next day exam on mycology. So again the bacteriology was conducted on the next day. Like this, the exams will be conducted in the simultaneously, classes also will be handled. And at the end of the week, this is important, every week end, whatever the topics that we have finished, for example we have finished 7 topics, on that particular 7 topics, there will be exam on at the end of the week. So we can generally call this week test. So week test generally happens at 6th day of every week. For example, first 5 days we do have a classes. On the 6th day exam, Sunday rest. Rest in the sense you can prepare simultaneously. If there is any other doubts or if there is anything else, please let us know. We will be happy to help you. And general plant pathology, general aspects are different areas of general plant pathology, or I would say like what are the different groups of plant pathogen which causes the disease, like general introduction about plant pathology. So the overall view about the plant pathology where we will discuss. And regarding mycology, it is a very big one, but we have a shortcuts or short way of representations by concerning exam preparations. So introduction to mycology, reproduction methods, types of flagella, general characters, specialized structures, and all those things will be discussed today. In the continuation, fruiting bodies, taxonomy, in the classification, especially we're going to concentrate on cricketal classification. That was given by Cricketal in 2008. That is one of the important and 10th classification. And along with what all there in Cricketal class, protozoa, chromista, Oomycota, 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 and Oomycota was continued since it is a very big one. Maybe 3-4 days it will require. Again, Bacteriology another 2-3 days, and last one is anamorphic fungi. I have simply given the basic things here, but when we go in detail about each and every single thing, for example, you can take Oomycota. Oomycota itself is a very big phylum, right? So every single class, every single phylum, every single subphylum, every single order that we will discuss, especially in exam oriented. If you want to read this book, the mycology itself is a very big one, but we don't have that much amount of time since we are preparing for exams. So in exam oriented way, what are all the things that we are going to discuss that we will see in this in the question. Again, at the end of 3rd week or 4th week, there is a weekly test. 3rd week there is a test. 4th week there is another test. Another important thing is like monthly test. This is an evaluation test. Student who have registered as subscribed, their monthly evaluation test, by concerning this monthly evaluation test, we consider their presentation or their way of preparation, and if they are doing good, it's okay, well and fine. If they were little bit lack of basics, or still they are struggling to get the the basic kind of information, or the way what they are studying, that also will be suggested in a different methodologies, and all those things will be taken into consideration. That's why I told you only 50 members, because we need to take care of every single individual who have subscribed, right? So 50 members we can accommodate very easily, and we can take care by every single person name by name. The rest we cannot handle, that's why we told you 50. And the next one is in bacteriology. See, these are all discussions are especially related to exam oriented only. I'm telling you syllabus right now, but on the next there are 4 5 slides are there. How these classes are going to be like for our easy understanding. In this demo one, I'm just showing you the syllabus and what we are going to discuss. Second thing is like how this orientation is going to be like, how this how the classes are going to be like, especially in a exam centric or exam oriented. And when it comes to bacteriology, we'll discuss about recombination techniques, growth curves, and the general morphology about the bacteria, pigmentations, antibiotic produced by bacteria, classification of antibiotics is a very important one, and entry methods of plant pathogens, because how this bacteria enters into the plant pathogen, the mode of entry, Fastidious vascular bacteria. This is also bacteria, but different groups of plant pathogen which generally causes diseases. And along with the endospore, secretion systems, survival and dispersal of plant pathogens, especially bacteria. What are the different kinds of phytotoxins that is produced by bacteria? List of important plant pathogenic bacterial diseases and the genera. Virus, like if there is any diseases that is associating with transmission of vectors, we can call it as the vector-borne plant bacterial disease, right? And other general topics, if there is any leftover topics for there in bacteriology, that will be discussed. At the end, there is every week there is a weekly test. Every month there is a monthly test, and the most important thing is every single day there is an exam. Whatever we are discussing, there is an exam. I will send you the question paper. And a couple of people are asking regarding the recorded videos. Of course, the videos will be recorded and uploaded to YouTube, but not everyone can watch the videos. We will provide you special link. Not everyone can watch, because we put the videos in a private, and by adding you, the people whoever subscribe, they will get a link. Through the link they can watch, even though the classes were missed, they can watch the videos. Other people cannot watch the videos because we are putting in private. Yeah. And regarding virology, general properties of plant viruses, morphology, viroids, classification of plant viruses, virusoids, prions, satellite viruses, mycophages, mechanism, infection, spread, transmission, virus vector relationship, all those things that we are going to discuss. Epidemiology, regarding epidemiology, disease triangle, factors affecting types of epidemics, pathogenesis is another important one, post pathogen interactions, remote sensing, computerized models, stimulation models, gene to gene hypothesis, survival and spread. If there is any remaining leftover topics as per ARS, ASRB given syllabus, that is also we will discuss. And again, weekly test every week, as I told you, there is a weekly test, daily test, at the end of the month, monthly test is also there. Yeah, regarding applied plant pathology, fungicides and classification, classification based on chemical nature, abbreviations like fungicide abbreviations, methods of application, especially the special methods of application included, the trade names of fungicide, and especially what this fungicide is especially meant for and what kind of diseases we can use it to manage the diseases. For example, you can take tricyclazole. This is especially meant for rice blast. Like that, every single fungicide, whatever I am telling you, or maybe trade names will be there. We'll also try to keep, what are the kind of diseases used to manage in a very easy way only, because we have different ideas to make you remember, because we also started our life from student, right? So we know how the students will feel. So don't worry about it, but we made, the pathology very easy for you. And next one is the antibiotics in plant disease management, mode of action of systemic fungicide, mycoherbicides, and idiom, and various components of idiom, and along with the weekly tests. And regarding diseases of crops, we have divided into two: field crops and horticulture crops. See here, we are not going to discuss every single crop symptoms management. No, exam oriented way. What are all the things we need to keep into consideration? Even though if you don't know, what are all the way to remember, just at least to guess, not to remember, to guess at least what might be the pathogen, what might be the management practice, what may be the specific fungicide, like that. In that way we will discuss. Some pathogens may be having specific symptoms, like for example, you can take rice blast, diamond shaped spots, or spindle shaped spots are there, right? So such kind of things. Again, they will produce pyricularia, pyriform conidia, like that, specific kind of bits are specific, very important information that we will discuss related to disease of field and horticulture crops. In fact, they are given simply like disease of crops. So disease of crops, in the sense, in agriculture we do study what? Field crops and as well as horticulture crops, right? So both will be included. And coming to the laboratory techniques and methodologies, this was a very important one for ARS also. We will definitely, dig deep into this laboratory techniques and methodologies as well as applied plant pathology also. Here we will discuss about glasswares and equipment, microscopic examinations or microscopic types of spores, preservation, maintenance techniques, and various other kinds of techniques in the detection of plant pathogens like blotting techniques, electrophoresis, PCR, LAMP, ELISA, CRISPR-Cas, and various other kinds of things we will discuss in this laboratory techniques and methodologies. Yeah, this is a very important one. Many people are, not good at this. Even me, once upon a time. Physiological and molecular plant pathology, where we will discuss DNA barcoding, hypersensitivity, elicitor, phenolics, phytolaxins, types of PR proteins, how these plant pathogens actually infect host pathogen interactions, toxins, enzymes that is produced by the plant pathogens, growth regulators, the mechanism of pathogenesis, hypovirulence, origins and types of origins, and along with the PAMP, DAMP, PTA, ETA, PRR, QTL, RNA. These are all some advanced techniques used, like how this plant is conferring resistance, or maybe how these pathogens are, continuous evolution. So the plant need to survive, so it evolves with having the genes or resistant kind of genes. How this pathogen is infecting, even though resistant genes are there, how again the plant is combating back. So all those things that we will discuss in physiological and molecular plant pathology. Later weekly test will be conducted and daily test also. And along with the Phenerium plant parasite, there are only 4 were there, and Cuscutal or the Swarbanchus triad, quarantine and biocontrol agents. So all those things that we will discuss in detail. At the end, if there is any leftover topics were there, if there is any required topics as per the rules of ASRB, ARS, or net SMS STO, that we will also discuss based on the subscribers wish. Discussions related to previous year net question paper 2006, 7, 9, 14, 1, 14, 2, 18, 1, 18, 2, 19, 21, 23, 23. And the most important thing is, by discussing these question papers, we will concentrate on the areas, what kind of areas they are placing, like general ideology and the general understanding. If there is any leftover topic we will discuss, and a daily exams test, evaluation test, as I told you, daily test will be there, weekly test will be there, monthly evaluation test will be there, and overall test will be there, grand test will be there, just a couple of days, maybe 15 or 20 days before. However, this net exam will be conducted. The same way we also will conduct your exam to train you exactly as per the net exam. So that will be like the last minute recap section. And the most important one is we prepared a PPT presentation which is over 250 slides, especially for the students. The last minute preparation, so they don't have time since the beginning. So such kind of people, we have prepared a last minute preparation. So that preparation is very much useful for the last minute preparations and all those things. That is also we're going to discuss in this training session. And this timetable what we have discussed slightly may changes or may slightly varies, not too much, slightly varies according to the time and the exams what we are conducting, all those things. Yeah, now entering into this demo one, this is a sample discussion, like how these classes are going to be and how we are going to train you and how this training is going to be like. So for easy understanding, I took 2 general topics. I may also took history or maybe other kinds of things. I randomly selected 2, that is generally Pythium and Pythophthora. Let's discuss about this and we see in a way of exam orientation. Our presentation is also pure and pure of exam orientation as well. Yeah, see, take a look at this. Pythium, generally Pythium means decay. Okay, so generally this Pythium generally causes diseases in nursery stages, right? Especially this disease was observed in vegetables mostly. Generally this Pythium causes a disease called damping off, right? So damping off is generally caused by, we mostly study a pathogen called Pythium aphanidermatum, right? So this Pythium produces a sporangia, which is a globosporangia. This is like round or globose kind of sporangia, which is non-papillate. Again, there will be a new terminology. What is papillate? What is non-papillate? What is semi-papillate? All those things that we will discuss. So papillate in the sense there will be a small kind of this kind of outlook, outer structure will be, that is called papillae. Semi-papillae means half. No papillae or non-papillate means there is no papilla above the sporangia. This is a very important point. Another thing is, so generally this Pythium produces oospores as a sexual spores, zoospore as a asexual spore. And the type of antheridium, because the type of reproduction in Pythium is like if you see gametangial contact type of sexual reproduction, like so gametangial contact type of sexual reproduction where male and female come in contact with each other and male gametangium passes to female gametangium, fertilization takes place. So generally another point is the type of reproduction in Pythium is gametangial contact type of sexual reproduction. Majority of the Pythium species are homothallic. They consist of only one kind, right? Homothallic. So for this management of Pythium diseases, another one more point I have mentioned here. So like this there is a globo structure that is formed above the sporangium of this Pythium, that is called vesicle. So which means in exam they will ask, so the formation of vesicle can be observed in which life cycle, especially Pythium. If you take Phytophthora, there is no vesicle is formed above the sporangia. Yeah, and there is no other fungi which is produces a vesicle on the sporangia. So the formation of sporangia is a special character of Pythium. So Pythium produces vesicle above the sporangia. That is another important point. The specific fungicide, what are all the specific fungicide that is recommended for Pythium is metaloxyl, generally very important one. For all Oomycetes or Oomycota group of fungi, we do recommend a specific fungicide called metaloxyl. The trade name is Ridomil. Since we are MSc, we need to study this trade names also. If the same metaloxyl is combining with the mancozeb, the trade name is Ridomil gold. If it is a Ridomil gold, it is a combination fungicide. Metaloxyl plus mancozeb. Mancozeb may be 63%, metaloxyl may be 8 to 10%, I think so. So versatile aluminum, these are all some specific fungicide that is recommended for this management of Pythium diseases. And what is this Bordeaux mixture? We might have studied somewhere else, right? Yes, of course. There is a specific fungicide called Bordeaux mixture. Very important one. What is this Bordeaux mixture? The remaining other points we might have studied there. So let's discuss about this Bordeaux mixture again in the later slides. But if you see this presentation, what I have given here exactly represents the exam oriented. As I told you, paragynous. This is a paragynous, which means antheridium and oogonium, which lies side by side. Antheridium and oogonium, which lies side by side. It is called paragynous. Para means side by side. So that is called as paragynous antheridium. So these are all the important points we consider into for Pythium. Generally decay, so which causes damping of diseases, that is Pythium ophanidermatum. The sporangia is globose, which is an anpapillate. Sexual spore is oospore or asexual zoospore. Type of reproduction gametangial contact. Paragynous type of antheridium, which means antheridium and oogonium which lies side by side. And another thing is the formation of a vesicle in the life cycle can be observed in Pythium only. Specific fungicide metaloxyl plus mancozeb, Ridomil gold. Sometimes they will ask trade names also. And this Bordeaux mixture we will discuss. Just take a look into in exams how they will ask. Which of the following pathogen produces paragynous type of antheridium? Or most of the Pythium species are homothallic, heterothallic, both homo and heterothallic, none of the above. Like this, they will ask questions. That's why I have given exactly exam pointed, I have exam centric points I have given here. Yeah, we discussed Bordeaux mixture for this Pythium, right? So anyway, so whatever all the new points that are coming up, we will also discuss and we will take the notes. For example, if you are studying something, what are the new points or adding points or side points that will come to you, there itself we will take a clarification about each and every single aspect and point. Generally, I am telling how these classes are going to be like. For example, Bordeaux mixture, as I told you, like generally used for the management of damping off also. For damping off, we can also use Bordeaux mixture, but generally metal oxide is the specific fungicide. Okay, so let's discuss about what is this Bordeaux mixture and all the history and mystery. Okay, so this Bordeaux mixture generally called as holy water. This was developed by a scientist called Millardet in between 1882 to 1885. This is a very specific fungicide, especially meant and developed for a disease called grapes downy mildew. This is one of the very important disease that happened in France. It is a epidemic in France in 1878, and the pathogen is Plasmopara viticola, which causes grapes downy mildew. Especially for the management of grapes downy mildew, Millardet developed this Bordeaux mixture, and the composition is 1:1:100. What is this 1:1:100? Here 1 kilogram of copper sulfate, 1 kilogram of hydrated lime, 100 liters of water we can call as Bordeaux mixture. Copper sulfate, lime, and 100 liters of water is Bordeaux mixture. So, but the originally the composition given by Millardet is for Bordeaux mixture is 5:5:50, which means 5 lb of copper sulfate, 5 lb of hydrated lime, and 50 gallons of water. If you convert the same way into this also, it will come close to 1:1:100 only. For easy understanding, the formula slightly changed to 1:1:100. But original composition that was given by Millardet is 5:5:50, which means 5 lb. 1 lb equal to 2.26 kilograms. 5 lb means around it will come, 1 lb equal to 1 lb 0.4 kilograms. So 1 lb equal to 0.4 or 0.45 something it will come. 5 lb means it will come close to 2.26 kilograms. So 5 lb for 2.26, 50 gallon. Each gallon equal to 3.8 liters of water. So if you just see 2.26 into 3.8 under 180 liters are that way, again it will come close to 1:1:100 only. For easy understanding and remembering, the formula slightly changed to 1:1:100. Now we have a couple of points related to Bordeaux mixture. Holy water was first discovered by Millardet 1880 to 85. Generally you can consider as 85. Used for the management of grapes downy mildew, Plasmopara viticola, which is the epidemic in France 1878. Composition is 1:1:100. Like that we remember, right? And just related to Pythium, there are a couple of important diseases. In exams, they will also ask like foot rot of cucurbits is caused by foot rot of papaya. This is a very important one, and a couple of times this was asked like that. Okay, damping off of vegetables. These are all some list of important diseases. Pythium aphanidermatum, foot rot of papaya, Pythium aphanidermatum, foot rot of rhizome rot of ginger, aphanidermatum as well as myriotalium, foot rot of cucurbits, Pythium aphanidermatum, Pythium butleri, damping up of mustard, Pythium mammalium, damping up of angiosperm, generally aphanidermatum, de Baryanum, ultimum, or irregular. So related to Pythium, I have given a couple of points. So those are all exam oriented points along with the list of important diseases I have given. Coming to the next one, Phytophthora. There is a lot to discuss about this pathogen, which is responsible for Irish famine. In Ireland in 1845, there is a disease called Irish famine, which is the lead blight of potato, which is caused by a fungal pathogen called Phytophthora infestans. So the name Phytophthora means Phyto means plant, Thora means destroyer. In Greek, Phytophthora means plant destroyer. Why it is named as Phytophthora? Because it is destroying the entire plant once it gets infected. So it is named as Phytophthora. The name Pythium was given by Anton de Bary. Later, I will tell you about the Anton de Bary. Okay, this is actually a well-suited name because of their destructive nature. The name Phytophthora was given by Anton de Bary in 1861. So before giving the name Phytophthora, this late blight pathogen was called as a Botrytis infestans. This name Botrytis infestans was given by Montag and Berkeley in 1845 and 46 respectively. So, however, Pythium produces a paragynous type of Anthracium. This Phytophthora produces amphigenous type of Anthracium. Paragynous means where Anthracium and Oogonium which lies side by side. When it comes to amphigenous Anthracium, you see here, Anthracium is down. Above the Anthracium, Oogonium is there. Okay, na? So above the Anthracium, if the Oogonium lies This is called amphigenous type of Anthracium. Generally in the life cycle, if you see this oogonium, which actually passes through the Anthracium and they form a small globo structure above the Anthracium. This is called paragenous type of Anthracium. This is a very important one. The Phytophthora pathogen produces the type of Anthracia the Phytophthora pathogen produces amphigenous type of Anthracium. So what is the first name of lead blight of potato? Botrytis infestans. Phytophthora means plant destroyer, plant destructor, plant killer, something like that they will ask. Okay. So Phytophthora means plant destroyer. The name Pythophthora was given by Anton de Bary in 1861. Before giving the name Phytophthora infestans, actually the life cycle was discovered, the pathogen was discovered in 1861, but the pathogen name was given by Anton de Bary in 1876. Sorry to confuse you. Anyway, so 1845 the disease occurs. 1861 Anton de Bary identified this is a fungal pathogen, and the name Phytophthora was given by Anton de Bary in 1876. 1845 lead blight of potato. 1861 identified a pathogen and the life cycle of the pathogen by Anton de Bary, and the name Phytophthora was given by 1876. That is also given by Anton de Bary only. Okay. So before giving this name Phytophthora infestans, the original name of the pathogen is Botrytis infestans, and this Phytophthora produces amphigenous type of Anthracium. Amphigenous means oogonium which actually lies are present above the Anthracium. This is called amphigenous type of Anthracium. So there the pathogen produces globosporangia. Pythium produces globosporangia, but a Phytophthora produces lemon-shaped sporangia. This is exactly look like a lemon shape. Okay, somewhat lemon shaped. Okay. So there Pythium pathogen non-papillate, where this Phytophthora pathogen produces papillate. Above there is small structure, some kind of structure that is called papillate. Okay, for production of Phytophthora, again same, Pythium and Phytophthora produces sexual spore as oospore, asexual spore as zoospore. Okay, here the zoospores are produced around the temperature of 12 to 15, whereas the gem tube development takes place around 18 to 20 degrees Celsius. And from the beginning of this Phytophthora, we are talking about a scientist called Anton de Bary. Who is this Anton de Bary? Let's see. Yeah, Anton de Bary is generally considered as the father of plant pathology, or we can also call as father of modern plant pathology because of the contributions he have done. He is also called as father and founder of modern experimental plant pathology. Why is called experimental plant pathology? So before proving this lead blight of potato, there is no such experiments have been existed. He performed a very small pot culture experiment to prove this late blight of potato is a fungal origin. So he just initiated this conducting experiments, right? Experiment, right? So that's why he called as the father and founder of modern experimental plant pathology. He wrote a book called Comparative Morphology and Biology of the Fungi, Mycetiosa and Bacteria. So there is nothing to confuse. We are discussing just two demo topics I took, Pythium and Phytophthora. In Phytophthora, I'm just telling you exam-oriented points. We have discussed about Anton de Bary. Then you can ask me. So we are discussing about Phytophthora. We can discuss and finish it, right? So anyway, what are the connecting things? As I told you, Anton de Bary, we might have studied Anton de Bary somewhere else. What's wrong if we recall, right? We will recall every single thing, no problem. We will remember, and it will be very easy for recall when the exam is happens, right? So the book is Comparative Morphology and the Biology of the Fungi, Mycetiosa and Bacteria. He introduced many terminologies, many new terms what we are using that was introduced by Anton de Bary, like Astoria, otherwise called as sinkers. Symbiosis, we know very well, mutual beneficial relationship, right? Symbiosis, that is also coined by Anton de Bary. Teleospores or teletospores, Mycetiosa, that is protozoa. For protozoal disease called as Mycetiosa. Okay. Sex hormones, the first sex hormones, iron and anthraciol. Autoecious and heteroecious nature of rust. Chlamydospores. Another point is endophytes. Endophytes is also coined by this Anton de Bary. And he have trained, I would say, some kind of PhD students or maybe students. He have trained a couple of students, many students he have trained, and some of them become the greatest contributors of plant pathology. For example, is you can take MS Voronin, Oscar Brefield, Millardet. Just now we discussed Millardet, right? HM Ward, WG Farlow, Fisher. These are all are the students of Anton de Bary. Let's see, we have discussed the students also. Let's just take a look into the basic understanding about this and who are all these scientists and what are all their contributions. MS Voronin discovered the life cycle of clubroot of cabbage. What is this clubroot? Very destructive and severe disease in cabbage, or crucifers, which is caused by Plasmodium porobranski, which is a protozoal disease. The life cycle was discovered by MS Voronin. This life cycle of clubroot pathogen produces two main kind of symptoms. What are all they? Hypertrophy and hyperplasia. For the management of disease, it is not necessarily to go with any fungicide, just raise the soil pH, because this pathogen is highly favored by lower soil pH around 5.2 to 5.5. If we raise the soil pH around 7.7 or 7.8, above 7, we can control the disease. What we do for the raising of soil pH? We do generally apply lime. Okay, so in that way we will read. So Oscar Breffield generally developed a pure culture technique and single spore isolation, and is also the first person who introduced gelatin as a solidifying measure in microbial cultures. Millard, just now we discussed, developed Bordeaux mixture. The composition is 1:1:100, which is recommended for the management of grapes downy mildew. Grapes downy mildew is caused by Plasmodium viticola. Like that we will study. Okay. H.M. Ward, father of tropical plant pathology, and actually the one who discovered this uh, very important role in the identifying the reasons behind this coffee rust Camellia vastatrix, and is also worked on identifying the life cycle. Somewhat he have done, but another important contributions are done by other scientists. And W.G. Farlow, first independent course of plant pathology in Harvard University was started by W.G. Farlow only. These all are the students of Anton de Bary. Here 2 points are there. First point, I might have left or stick to only the particular topic what I am discussing, or else we will discuss every single new thing that is coming, just like remembering the things. This is a very easy way, in fact little bit laborious way, but still if you ask me, this is a very easy way to remember the topic and recall the topic. Yeah, Anton de Bary, he's Anton de Bary. These are all the important terms he introduced: astoria, tiltospores, heterosis, symbionts, six hormones, obligate parasitism, endophytes. So these are all are the things that was discovered by Anton de Bary. Yeah, continuation to this Anton de Bary, he discovered the life cycle of Phytophthora infestans, that is the late blight of potato, which is responsible for Irish famine. I am telling once again, late blight of potato that strike Ireland in 1845. Before that also the disease was there, but that is not that much severe. Forget about it. 1845, late blight of potato, Irish famine. 1861, Anton de Bary discovered the life cycle and told that this pathogen is caused by a fungal origin. He named the pathogen as Phytophthora in 1876 only. The name was given in 1876 only. Up to 1876, the pathogen was actually called with the name of Botrytis infestans, that was first given by Montag 1845, Berkeley 1845 and 46. And he also demonstrated the heterogeneous nature of rust fungi. And first physiological work on plant pathology, that is especially Botrytis cinerea, he is the one who has done. And he's also studied or reported the role of enzymes and toxins in the direct penetration, for disintegration of tissues or for killing of tissues also he have studied. And first work on Sclerotinia sclerotianum in 1866, this tissue disintegration. And he named the pathogen Phytophthora. Phyto means plant, Thora means destroyer. The name Phytophthora is also given by him only because of the destructive nature of the pathogen. And both Pythium and as well as Phytophthora, they are Oomycota group of fungi, Oomycetes. Okay, this is not a true fungi. We can also call as Comista group. Okay, in classification, there are main three branches are there for related to our plant pathology. We do study all the time: Protozoa, Comista, and fungi. This Oomycota is not a true fungi. It is a fungi-like organism. That's why it is actually placed under Comista. Okay, this Phytophthora and Pythium both are Oomycota group of fungi only. Generally, Oomycota group of fungi cell wall is made up of cellulose and hydroxyproline. We also need to remember this hydroxyproline because in 2021 and 2023 NET exams, we'll also discuss the previous year question papers just before a couple of days before exams. Here they have asked in 2021, Oomycota cell wall is made up of. They have not given cellulose, but they have given hydroxyproline. So which means they consist of both cellulose and hydroxyproline. Then I have gone through why they have given this hydroxyproline instead of cellulose, because the majority of the time we do study cellulose also. If we have searched in detail and went deep into it, then I realized hydroxyproline is also one of the major component of this Oomycota cell wall structure for maintaining the integrity. So this hydroxyprone also plays a major role. That's why they have given that also. Like in exams, they will ask anyway. We will study and we get ready. We'll make ourselves ready for all the directions. That's why I'm telling you. In this way, we will study. Okay. So Pythium and Pythophthora Oomycota group. Generally, Oomycota group of cell wall is made up of cellulose and hydroxyproline. And generally, for all Oomycota group of fungi, we do recommend a specific fungicide called metaloxide. So which means for all Oomycota, we do recommend a metaloxyl. Yes, I think metaloxyl is going to ban. I think already banned also. So if the Oomycota group of fungicide is made up of cellulose and hydroxyproline, what about the fungal group of fungi group cell wall is made up of? So the fungal cell wall is made up of chitin. Okay, the fungal cell wall is made up of chitin, whereas Oomycota group of cell wall is made up of cellulose plus hydroxyproline. So this way we will study and we remember the things that will be very easy for us. Yeah, these are all a couple of important list of diseases that is caused by this Phytophthora. In rubber, abnormal leaf fall of rubber that is caused by Phytophthora palmivora, red gram stem blade Phytophthora dressleri pharma species kajani, gingerly leaf and stem blade Phytophthora parasitica, castor seedling blade. This is also very important one. Seedling blade of castor Phytophthora parasitica, tobacco black shank Phytophthora parasitica varnicotianae. And these are all the list of diseases that is related to Phytophthora, which means we have sorted out what are all the important points that is required for preparation. We have given very easy understanding. This is just a demo only. Demo will be like this, but just imagine how the classes are going to be. We do concentrate on every single aspect in detail. So the basic understanding you will get it. You will get it about what are all the overall understanding about plant pathology that we will assure you. Yeah, now we will see the difference between Oomycota and true fungi. So generally Oomycota generally called as egg fungi or pseudo fungi, whereas true fungi Ascomycota, Ascomycota and Bacteriology, true fungi. Cell wall is made up of cellulose and hydroxyproline. Just now we have discussed Oomycota, whereas the true fungi cell wall is made up of chitin. Another important point, Oomycota cell wall storage organ or food storing organ is mycolaminar, whereas the true fungi glycogen or I. Like these points are there, right? We have sorted out and we will discuss. Yeah, we have discussed two topics, right? Pythium and Phytophthora. Now we will see the overall easy understanding. Pythium means generally decay, Phytophthora means plant destroyer. So Pythium belonging to Pythiaceae, whereas Phytophthora belonging to Peronosporiaceae. Order Pythiales, order Peronosporales. Now this Pythiales also comes under this Peronosporiaceae. Astoria and Aprosoria generally absent in Pythium. When it comes to Phytophthora, it is present. Couple of important points are the very important points what we need to take into consideration. Such kind of things we will discuss. For example, if you see type of Antheridia, we have discussed Pythium consists of Antheridium is a paragynous, where male and female side lies side by side. When it comes to amphigynous type of Antheridium, this is Phytophthora, where this above male female lies, that is called paragynous. That is paragynous specially observed in Phytophthora. So in exams, they will ask anyway like amphigynous type of Antheridium is observed by. The formation of a vesicle above the sporangium can be observed in. So formation of vesicle can be observed in only in Pythium. In Phytophthora, there is no such kind of vesicle formation takes place. That is also important point. So this is just an exam oriented way. This is how the training is going to be like. Just a general introduction. So we'll also see what are the previous year questions that was asked, especially on Pythium and Phytophthora. We'll see. In NET 2023, there is a question called cashew damping off is caused by. I have mentioned list of important diseases of damping of disease, right? So generally, this cashew damping off is caused by Phytophthora. Majority of the time, damping off is caused by Pythium only, but some damping offs which is caused by Phytophthora also. Okay. Buckeye spot of tomato is caused by, which is caused by Phytophthora. In the list of important diseases of Phytophthora, I have given. Oomycota specific fungicide we have discussed, right? For Phytophthora, Pythium, all are Oomycota group of fungicide only. For Oomycota group of fungicide, we do recommend a fungicide called metaloxyl, right? And example of amphimobile fungicide, metaloxyl only. In 2021, they have asked. Amphimobile means both acropetal and basipetal, which means it has both directions. The action is both directions. So that's why it is called as amphimobile. Both directions it is moving. So it is called as amphimobile. Example is metaloxyl. This question was asked in 2021. And another couple of questions like Bordeaux mixture composition that was especially given by Millard, 5:50, 5 lb of copper sulfate, 5 lb of lime, 50 gallons of water. This is 2019 question. Phytophthora romorum causes which disease? Sudden oak death. In the list of important diseases of Phytophthora I have given, sudden oak death is caused by Phytophthora romorum. Is a 2019 net, and a favorable temperature required for juice spore and as well as sporangia germination. For juice spore, the Phytophthora requires 12. For the sporangia germination, the pathogen requires 24. This is 2018 net 2. Cenocytic mycelium, or we can also call it as a non-septate mycelium, can be observed in the genus Phytophthora. Both Pythium and Pythophthora, they will produce cenocytic mycelium, which means the mycelium consists of no cross walls. Without cross walls, the mycelium will be there. That's why it is called as cenocytic mycelium. So here we have discussed the important points. What are the probability question, exam-oriented way of discussion, all those things will be discussed, right? And in the demo too, I want to concentrate. I'm going to concentrate specific in detail about a particular topic. I'm randomly selecting the topic since it is a demo, right? So once we started the classes, I do conduct the classes on based on the syllabus, based on the order and the regularity. Since it is a demo, I have just took two topics, how this training is going to be for easy understanding. Yeah, I have mentioned already, this is our services for various kinds of things, detailed benefits and course structures and all those things we have published already every day. And I also representing the link here and also giving the link in the subscription, sorry, in the comment section. You just take a look at it. For further details, guidance, registration and payment, so anyone can reach us at researchergeeky@gmail.com, or else also the given number. And the net demo classes, as I told you, 11th and 12th, first class is already finished. There is only one day left tomorrow. And net classes starts from 21st/5/2025, which means exactly on one day before of the closing the registration, we are going to start the net classes on a daily basis. If you have not yet registered, you can register by seeing this demo class. If you have any doubts, you can feel free to reach us to the email ID or to the given number. And anyway, your subscription is definitely a promise from our side. We will definitely ensure the worthy every single moment, but not a missed opportunity. This is a promise from Team Geeky Researcher. Then why waiting? Let's get geeky. Yeah, for further details, students can reach us at researchergeeky@gmail.com. Stay geeky and stay tuned. We are Team Geeky Researchers.